Hello. I'm assuming you can all hear me. If you can't hear me, please let us know in the chat. My name is Jenny Rappaport, and I have a few intro remarks to make while we wait for people to finish trickling in. Welcome to Family History Today, the Center for Jewish History series of genealogy-themed public programs. My name is Jenny Rappaport, and I'm the head genealogist at the Ackman and Ziff Family Genealogy Institute. I will be your presenter for today, and I wanted to thank you all for joining us. Nobody has said they can't hear me, so I'm assuming you can. And I will continue onwards. The Center for Jewish History provides a collaborative home for 5 in-house partner organizations that together form the largest and most comprehensive archive on the modern Jewish experience outside of Israel. In addition, the center houses the Genealogy Institute, where we assist people of all ages, abilities, and levels of experience, whether Jewish or non-Jewish, in researching their family history. We encourage you to visit our Genealogy Institute on Monday from 9:30 a.m. to 4:30 p.m., no appointment needed. If you are a Holocaust survivor or the direct descendant of a Holocaust survivor, we are open Tuesday through Friday by appointment only. At the Genealogy Institute, you'll enjoy free access to genealogy databases and reference books with our genealogists and experienced volunteers on hand to provide guidance and direct you to relevant resources in our partner collections. If you're unable to visit in person, you may email us at gi@cjh.org to ask for advice on your research questions or to schedule a free 45-minute Zoom consultation. Okay. And I have a few housekeeping notes before we start. Please send us your questions and comments anytime during the program by using the Q&A box on the bottom of your screen. My colleague, Moria Amit, will be monitoring them. Please note, however, that I will only answer questions after the presentation in the dedicated Q&A portion of this program. If you'd like to view the captions for this program, click on the closed caption or CC button on the bottom of your screen and then click Show Subtitles. This program will be recorded. Please note that we do correct the auto captions for all of our program videos. So it typically takes us about 2 to 3 weeks to post them online after we do a Zoom program like this. Once the video is online, you'll receive an email with the link to watch it if you'd like to see it again. Okay. Bear with me for 2 seconds while I set up screen sharing because Zoom hates me. Okay. And eventually this bar up here will disappear. Okay, so today's presentation is on interpreting your DNA test results for Ashkenazi Jews. Actually, I am going to shut my video off while we're doing this to be less distracting, so you can just concentrate on Zoom. Actually, we're not. We're just going to talk. Okay. When you take a DNA test as an Ashkenazi Jew, your DNA results may look like this. These are mine. It's an excerpt from mine from Ancestry. As you can see, the green shows you that I am a perfect circle of 100% Ashkenazi Jews. Sometimes they try to give you little bits and pieces that say ancestral journeys. And then it gives you a very large number of DNA matches. This is from a couple of months ago, so I have more than this now, but it's 224,813 people, it says, are my relatives. And it's very overwhelming if you've never looked at a DNA test before. So in general, DNA results vary by database. If you have 4 Ashkenazi Jewish grandparents like I do, you will generally have 97 to 100% Ashkenazi Jewish reported as your ethnicity. You will have very large numbers of DNA matches in a commercial database. Even databases that are smaller, you'll still have tens of thousands of matches. And most of these matches are not.
Jenny?
Yes?
You're not showing the DNA slides.
Oh,
It's currently on the Anne Frank and
oh,
other programs.
my apologies. I will go backwards and do that again. Give me 2 seconds to go yell at Zoom quietly. Okay. Can you see DNA slides now?
Yes.
Wonderful. I apologize for that. I normally use Zoom on a MacBook and it works differently than it does on the machine I'm using at work, which is a Windows computer. Okay, so hello again, interpreting your DNA test results for Ashkenazi Jews, part 2, or take 2 rather. Your DNA results usually look like this. Like I said before, I'm a perfect circle of green for ethnicity. It shows me that I'm 100% Ashkenazi Jewish as my ethnicity, and it gives me a really large number of DNA matches, which is currently, at the time I took this screenshot, 224,813. I think I'm above 225,000 at this point because it was a couple of months ago. In general, DNA test results will vary by database, but if you have 4 Ashkenazi Jewish grandparents, meaning both your mom and your dad were fully Ashkenazi, you will have 97 to 100% Ashkenazi Jewish reported as ethnicity. You will have large numbers of DNA matches in any commercial database, usually on the tens of thousands or hundreds of thousands. And most of these matches will not be related to you in a genealogical time frame, meaning we can't trace them with a historical document. We can't show with like a birth certificate that this is your 17th cousin. And that's because of endogamy. So there are 3 keys to understanding your DNA results. The first is you need to sort of understand the basic science behind DNA inheritance. You then also need to know what endogamy is and the historical reasons for it. And finally, you need to see how endogamy has affected your DNA results. So DNA basics. DNA is deoxyribonucleic acid. It's the infamous molecule that's a double helix. You can see a picture over there from the US National Library of Medicine. It's 4 base pairs. Adenine and thymine always go together, and guanine and cytosine always go together. And it encodes the genetic information for how organisms function and develop using a sequence of the 4 base pairs. So when you look at raw DNA data, they will read 1 strand of the double helix, and it will be represented with a string of letters that look like they make no sense, like A, T, A, C, T, G, etc. Humans have 23 pairs of chromosomes, which are the structures that store our DNA. On the left, you can see a karyogram, which is a stained picture of somebody's chromosomes. That's what they look like when you lay them out. The DNA and the chromosomes that store the DNA are present in the nucleus, which is the central part of a cell, of every cell in your entire body. So it's in your skin cells, it's in your blood cells, it's in your heart cells, even though you're never going to take a DNA sample from a heart cell. You have 22 pairs of autosomal chromosomes, and then you have 1 pair of sex chromosomes, which is what you can see in the very bottom row in the middle, the X and the Y chromosome. You inherit 1 chromosome in each pair from your mom and 1 chromosome in each pair from your dad. Okay, and now we get to talk about meiosis. So meiosis is the biological process by which cells divide to form gametes. Gametes are the sperm cells and the egg cells. And it's the reason why you only inherit half a chromosome from each parent. Every gamete, so every sperm cell and every human egg cell, only contains 1 copy of each chromosome. There are no chromosome pairs present in egg or sperm cells. You only get that 1 single half a chromosome from each parent. And we're not going to go into the entire meiosis process, which, if you like biology like I do, is fascinating, but it's also really complex. But what we are interested in as genetic genealogists is the crossing over that occurs during meiosis. So crossing over is illustrated by the pink and purple chromosomes in this diagram. During meiosis, chromosomes go basically crazy duplicating themselves. So instead of having 1 of a pair of chromosomes, they make an identical second pair of chromosomes in each of the cells that are going to divide to form a gamete. And then, as you can see in the second step of the diagram, the pink chromosome and the purple chromosome decide they would like to swap pieces with each other. So the 2 chromosome pairs swap the pieces with each other, and they will eventually split apart, so you will only have one half of each of those chromosome pairs, but they will have traded information with each other during the meiosis process. And this is really cool because it's why humans and all living creatures have genetic variation, because during meiosis, we cross over and switch pieces of our chromosomes. And in reality, in practice, in genetic genealogy, what crossing over looks like is very similar to this diagram on the left. This is chromosomes 6 through 11, and it shows the DNA that I share with my maternal grandmother. All of the blue segments are where my DNA matches my maternal grandmother's DNA. So we have the same exact DNA in those same places on our chromosomes. But the gray bars indicate where we don't match. Since I've inherited this set of chromosomes from my mother, by process of elimination, all the other DNA on them must be from my maternal grandfather. So when my mother's DNA combined and formed the egg cell that eventually would become me, these crossovers happened. And they happen at the points where the colors change on this diagram. Okay, DNA inheritance 101. You'll inherit 50% of your DNA from each parent because that's how human reproduction works. The DNA segments that you inherit from each parent will be a mix of DNA that your parents inherited from their parents. So if we go back, you can see the blue pieces were what I inherited from my maternal grandmother. And the gray pieces were the pieces I had inherited from my maternal grandfather. So crossing over lends an element of randomness to the whole process and contributes to genetic variation across the human species. This is also why siblings can have the exact same parents, but inherit different DNA segments from each parent. So the average percentage of DNA that you share with your relatives. You will share 50% of your, this only, to my annoyance, marks the paternal relatives, but it's the same thing for the maternal line too. So you will share 50% of your DNA with your parents. You will also share 50% of your DNA with your siblings. And then as you get further, and you will share 50% of your DNA with your children. You don't necessarily share the same 50% with each of these people. And it goes onward as you expand outward. So if your sibling has a child, you share 25% of your DNA with your niece or your nephew. Similarly, you share 25% of your DNA with your grandparents. And if you go further back in time, you'll find that you share approximately 12.5% of your DNA with your great-grandparents and approximately 6.25% of your DNA with your great-great-grandparents. And similarly, it goes down too. So with first cousins, as you can see on the chart, you share 12.5% of your DNA. And then when you go one generation further, so for example, your first cousin has an adorable baby. With that adorable baby, you only share 6.25% of your DNA. And so on. Until finally, if you go really far down, you see third cousin twice removed, which is a third cousin 2 generations away from you. And you share 0.195% of your DNA on average. This does vary. The numbers have a little bit of wiggle room in them. Okay, we can express the amount of DNA we share with someone as either a percentage, like in the prior chart, or as a centimorgan measurement. Centimorgan is a unit that measures how frequently genetic recombination, like crossing over, might occur in a particular DNA segment. And what it's used for in genetic genealogy is as a rough substitute for the length of a shared DNA segment. For non-endogamous populations, so not Ashkenazi Jews, the greater the amount of centimorgans you share with the DNA match, the more likely it is that you are related to the match. So this is the same chart as the one I showed you before, except instead of showing percentages, it breaks it down and shows you centimorgans you might expect, the amount of centimorgans you might expect to share with each relative. And as you can see, let's see if we can see the mouse. So for a parent, the top number here is 3,485 centimorgans, which is about 50% of your DNA. But you'll also see there's a range included below. And the range says that it's totally normal with a parent to share anywhere from 2,376 centimorgans to 3,720 centimorgans. And this chart comes from the Shared Centimorgan Project by Blaine Bettiger. It's hosted on DNApainter.com, and it's really wonderful because it has an actual input box, and you can input the amount of centimorgans you share with a match. So if you have an unknown match and you share like 133 centimorgans, and then it will let you it will give you the probabilities for which of this variety of relatives, and this is only half the chart that I included on the slide, might be related to you with that amount. It works with endogamous populations to a degree, particularly with the higher order relatives, so a grandparent, a parent, a sibling, or the child of a sibling. It gets a little wonky when you go further away because it's more geared towards non-endogamous populations. So now we get to my favorite part, which is the endogamy. And in order to understand endogamy, you have to understand Jewish history. So this is a brief overview of Jewish populations. Jews have gone through several diaspora events over the last 2,500 years, which have dispersed them throughout the world. This resulted in distinct ethnic subpopulations of Jews that can be classified by geographic location. So Ashkenazi Jews are from Central and Eastern Europe. Sephardic Jews were originally from the Iberian Peninsula, and Mizrahi Jews are from the Middle East and North Africa, with some populations of Mizrahi Jews never migrating during diaspora events. And this is a lovely map, and the map breaks it down and shows you that where the dark blue is, that's mostly where Ashkenazi Jews can be found. The Sephardic Jews shows you both the Iberian Peninsula here of Spain and Portugal, but it also shows the places where the Sephardic Jews spread throughout the Middle East and the Mediterranean. The green Jews are the Mizrahi Jews, and then there's other additional yellow groupings, which we are not touching on in this talk. Okay, what is endogamy? According to the International Society of Genetic Genealogy, endogamy is the practice of marrying within the same ethnic, cultural, social, religious, or tribal group. Endogamy has occurred all over the world. It's in populations as diverse as Ashkenazi Jews, the Polynesians, French Canadians, the Amish, and Native Americans. Pedigree collapse, which you may have heard of, and endogamy are not the same, and you can have both genealogical concepts in the same population. So I made a slide illustrating what pedigree collapse is. So pedigree collapse occurs when close relatives who share a common ancestor marry each other. So none of these people are real. It's all, names that are alliterative. But as you can see, there's Abraham Antman and Bessie Berkowitz here have a child, Arnold Antman. They also had a child down here, Alice Antman. Now Alice marries Douglas Dimmenstein. Arnold marries Claire Cohen. They each have kids too. Their kids are first cousins to each other because they're the children of their siblings. And then Alexander Antman and Denise Dimmenstein, who are first cousins, decide they would like to get married to each other, which is legal in some states. And they have a child, Annabelle. Annabelle has what's known as pedigree collapse because instead of having 8 independent great-grandparents, she only has 6 because with the red arrows, Abraham and Bessie are repeated twice because they were close relatives who married each other. And this did happen in Jewish populations, but it's not the only reason why we have endogamy. So endogamy, the reason we have endogamy is Ashkenazi Jews started from a founding population, about 350 people or so, who lived in the German Rhineland, which is the area around the Rhine River, between the 9th and the 11th century Common Era. Recent ancestors of that founding population had survived what's known as a population bottleneck, meaning an event happened that caused the population to narrow drastically, whether it was disease or being killed off or various other events that could happen that would cause loss of life. And so the recent ancestors most likely migrated from Southern Europe, but were of evenly mixed Middle Eastern and European descent. And we actually know from recent research, there was a paper done from Jewish remains from Erfurt, Germany, that by about the mid-1300s, they were pretty evenly mixed still between Middle Eastern and European descent, and you can break that out in the populations. So beginning in the late 11th century, one branch remained in Central Europe, in the German area, like the study participants from Erfurt, or not really participants, but the remains that were part of the study, and the other branch headed east, especially to Poland and Lithuania. In the succeeding centuries, until the early 20th century, Ashkenazi Jews married almost exclusively within their faith, and also predominantly within their local communities, amplifying the homogeneity of their DNA, meaning I married your cousin and you married my sixth cousin, and we kept marrying everybody's cousins. So what this means when you're looking at DNA results now in 2025 is that among Ashkenazi Jews, a third cousin DNA match, and notice in quotes, in a commercial DNA database is much more likely to be a false match, because the amount of shared DNA the two of you have is commonly due to the fact that you are related to them on multiple branches of your family. This leads to people sharing multiple small segments of DNA with each other, which are usually only 3 to 10 centimorgans in length, which they inherited from one or more distant common ancestors. These segments are not traceable in a genealogical timeframe. You cannot find the common ancestor. It is exceedingly difficult. I like to call them community DNA fragments because we pass so many of them around each other as Ashkenazi Jews. Okay, now we're going to go to talk briefly about ethnicity estimates before I get into how to actually analyze the DNA matches. Every company, and that includes Ancestry, 23andMe, Family Tree DNA, MyHeritage, and Living DNA, uses their own reference populations to estimate your ethnicity during the last 300 to 500 years of time using computer algorithms. It's not an exact science. They are basing it on science, but there is still inexactitude to it. The DNA testing companies are comparing DNA segments that they think indicate a reference population. So this segment indicates you're Irish, this segment indicates you're Polynesian, this segment indicates you're an Ashkenazi Jew, with your DNA segments. And they're making their own best guess about this. You should absolutely take any ethnicity estimates with a grain of salt, and you should look for documentary evidence during your genealogical research. So if somebody says, you're Jewish, but hey, you've got like a 5% chance of being Scottish, and you have never once heard of your family being from Scotland, try to look for proof of that in documents. Similarly, a lot of commercial databases like to give what's known as automated DNA match hits. I feel as a genealogist, these have to be taken with an extremely large grain of salt, because automated DNA match hints like ancestry throughline are based on user submitted data. If the data that's in the user submitted family trees isn't accurate, you won't get an accurate DNA match hint. The algorithm may think it is an accurate hint, which is why it's suggesting it to you, but it can't actually analyze whether the information in the family trees is correct. Similarly, Ancestry assigns what are known as paternal side designations, parent1/parent2, using their side view technology to as many DNA matches as it can. This works fairly well with non-endogamous populations. My husband's DNA is non-endogamous, and it breaks things out beautifully between his paternal and maternal side. This works extremely badly for people who come from an endogamous population, like Ashkenazi Jews, since your parents will often share DNA segments and DNA matches due to endogamy. Do not trust the parental side designations that have been assigned to you or endogamous DNA matches. They will likely not be accurate, and you'll see later on some of mine are marked with exclamation points because it's very angry at me. So what does endogamy look like practically in DNA results? It means you will often share more DNA with a known relative than someone from a non-endogamous population, because you've got all those tiny fragments of DNA adding to the total. DNA companies use various algorithms to try and eliminate the small segments of DNA that are due to endogamy. However, these algorithms vary by company and also by effectiveness. Ancestry has the Timber algorithm, which does not apply to your DNA matches if you share more than 90 centimorgans with a match. And you will find, as an Ashkenazi Jew, there is a significant amount of people from that like 200,000 you match that you share more than 90 centimorgans with. This can also lead to inflated totals of shared DNA with close relatives. So this is my dad's results. And you can see I'm at top. I come across as his daughter, and we share 3,430 centimorgans, which is about 50% of our DNA. And then these start to change. So this is my dad's paternal, this is his maternal first cousin. Ancestry insists that it's both sides. It's not. He shares 939 centimorgans with his first cousin, when on average people only share 866 centimorgans. This is his first cousin once removed on his dad's side, and he shares 501 centimorgans with her. and 433 centimorgans is the average. And it goes on with that. Sometimes it's a little less that you share with someone. And then when you get down to second cousin, this woman right here, we are not entirely sure how she is related to our family, although we do know she is related to our family. My father shares 238 centimorgans with her on her maternal side, and she is likely a second cousin once removed, although I have not yet fully proved that. On average, you only share 122 centimorgans with that type of cousin. And you can see it down here too. This is a known second cousin once removed on my father's side. And his maternal side. And on average, you only share 122 centimorgans. You can also see, it's very tiny on the slide, I apologize, this little red exclamation mark that Ancestry has given me. That is because I have assigned it to the maternal side, because I know this woman and have known her my entire life. And she is definitely a maternal cousin of my father's. And Ancestry insists that she can't be. And it gets very angry when you change the side designation. So you also get DNA matches that look closer than they are. These are 3 DNA matches of my father's. And you'll see they're marked with unknown woman, unknown man number 1, and unknown man number 2. None of these people, even though they share 105 centimorgans of DNA, are people that I can identify as relatives in a genealogical time frame. Because they share more than 90 centimorgans with my father, the timber algorithm is not applied. So within that 105 centimorgans are a bunch of smaller DNA fragments that are all adding up to look like it's a larger amount, and that they share more DNA and are more closely related than they actually are. So zooming in on unknown man number 1, when you look at any of these matches, and there's a plethora of them you will have, you can see that it is made up of many segments of DNA. This is 105 centimorgans across 11 different segments, where the longest segment of that is only 15 centimorgans. So that means there's 90 other centimorgans that are split between 10 segments, which is approximately like 8 to 9 centimorgans a segment. Some will be a little bigger, some will be a little smaller than that. A good indicator when you're trying to assess a DNA match is how long is the longest segment of DNA that you share with them. You would like it to be at least 20 to 30 centimorgans long for a match to be traceable by combining it with research with genealogical documents. And then you have matches that look more distant than they are, because DNA inheritance does have an element of randomness. This is a known second cousin once removed to my father. And we have a very strong paper trail going back several generations of documents, including in Poland, proving the connection, and the DNA segments they share also correlate with other known cousins in their family. But Ancestry thinks that the relationship is much more distant, partially because of how the timber algorithm had trimmed some of the DNA. So you can see it's showing shared DNA, which is what the timber algorithm thinks they actually share, which is 35 centimorgans across 6 segments. But their unweighted DNA is really, which means it has not been processed by the timber algorithm, is 57 centimorgans, which puts them more in range for their relationship. Timber has taken off 22 centimorgans of their DNA. And it will also give you suggested matches. So how do you analyze your endogamous DNA results, particularly if you test somewhere like Ancestry that doesn't have a chromosome browser? There are several methods you can use to do this. You can sort shared DNA matches. You can use the Leeds method. You can use the SMARTTA method, which always sounds Swedish to me, but isn't. You can build out family trees for your DNA matches. And you can also use things like tools on GenMatch. We're going to talk about each of these. So, sorting shared DNA matches. You need to buy the Pro Tool subscription to Ancestry to do this. Personally, I would look for when it's on sale, because they give you good deals. But if it's not on sale, it's about $10 a month. And it's an individual subscription, and you can pay by the month. So Ancestry Pro Tools gives you the ability to sort your DNA matches by how much DNA they share both with you and with another person who you both have as a DNA match. You can sort your shared matches by newest matches, by how much DNA they share with you, or by how much DNA they share with the other person. This lets you get a clear view of how you might be related to someone, especially when they don't have a family tree attached to their account. Remember, we're genealogists, we're detectives of some sort. So you're using the sorted matches to be a detective. By searching their matches, you may find they have a close DNA match who does have a public family tree, which you can then extrapolate from the information in it. So this is what shared DNA matches look like. This is my half first cousin, once removed. Is my mother's first cousin, so he's my half first cousin once removed. We are only related through my great grandfather. Initially, when you first go to the shared matches on the Ancestry Pro tools, this is what it looks like. And it doesn't give me very much information, especially because matches 1, 2, 3, 4, and 5 are likely not traceable for me. But once I sort my cousin's DNA matches by closest to distant matches, meaning how close they are to him and how distant they are to him, a pattern emerges. And all of a sudden you see this is his granddaughter over here, who is my half second cousin once removed. And Ancestry has, similar to the first example, trimmed much more DNA off and thinks that it is my half third cousin once removed. Similarly, this is my cousin's grandson. And then we go, this one I don't actually know, because I'm pretty sure they're on the other side of his family. But it becomes more pattern-like, and you can see relatives. Now, you can also see over here, this little family tree icon. If I didn't know who this person was, and the match didn't have a family tree, I could click on the tiny family tree icon, and I could browse their family tree, because if it's their granddaughter or their grandson, they're fairly closely related. And I can also use their closest matches to eliminate which side of the family those matches might be on for them, because I share very little DNA with them, like here, 19 centimorgans is nothing. Okay. So the Leeds method somewhat develops based on shared matches. And I will give you the caveat that it only works if you are only partially Ashkenazi Jewish. So what the Leeds method does is it uses your shared matches, who share between 90 to 400 centimorgans, to cluster together people who descend from a common ancestor. It was at first done with a spreadsheet. You may hear about auto clustering now with certain technologies. In fact, Ancestry is supposed to bring auto clustering out, and a button for it briefly appeared last week, did nothing, and briefly disappeared. So I'm assuming it's in beta testing. But basically, you can do it with a regular Excel spreadsheet or Google Sheets, and you would, if you are only partially Ashkenazi Jewish, meaning your mom was Jewish and your dad wasn't, or vice versa, or you have a grandparent who was Jewish and everybody else isn't Jewish, it's much easier to sort the matches by relative. And each color is the people who match you also match the same people. And so eventually you're figuring out what the common ancestors are between everybody. And the full directions can be found at genealogist Dana Leeds' website. The URL is here. But it doesn't work if you are from a 100% endogamous population. Because of endogamy, many of your DNA matches will show up as shared matches to each other. So even if you know they're a maternal match on your mom's side, they'll also show up as matching your dad. And this prevents you from clustering them very beautifully by ancestor, like on the spreadsheet. So to address this problem, Steve Taub, who is a Jewish genealogist, has developed the SMARTTA method, which stands for Shared Match Analysis on Rows with Top Tier Averages. And it's a big mouthful, but basically, the basis of the method is that it looks at the total centimorgan shared with a match, as well as the average centimorgan size of each DNA segment. And it involves math, basically. Unfortunately, you need to use math. So basically, we can take the total centimorgans that Ancestry gives you. So in this example of an unknown match that I have, it says I share 112 centimorgans with this person across 13 segments of DNA. And so you can divide the total centimorgan by the total number of segments. So when I divide 112 by 13, it comes out that each centimorgan segment is roughly 8.62 centimorgans. Now we know that one of them is 19 centimorgans, so some of them might actually be smaller than that. But on average, it's essentially a rounding error where endogamy is concerned, because the segments are going to be very small and they're likely going to have been inherited from multiple common ancestors. And it's likely to be unsolvable. The complete directions, which are complicated, but worthwhile to read, are found on Steve Taub's Medium post. And this chart is taken from that Medium post. And it shows what the SMARTTA method considers solvable matches. So it breaks it down, as you can see, into colors. So your relatives, it doesn't matter how many centimorgans they have or what the average centimorgan segment size is, you know who they are because you can already place them in your family tree. And then you start looking at unknown relatives. So if you have an unknown relative who shares at least 150 centimorgans with you, it's likely solvable to some degree, like the woman I pointed out, who we think is a second cousin once removed from my family, but we're not 100% sure yet. And it doesn't matter what the average centimorgan size is for that. And then you get to what's known as tier A, which is if you share between 90 to 150 centimorgans with a match, and your average centimorgan size is 15 centimorgans, which means you want, if you share 100 centimorgans with a match, you want it to be at least, you want it to be over 6 segments or less, because that'll give you about 15 centimorgans. Similarly, tier B is if you share 90 to 150 centimorgans with a match, and it's between 12 to 15 centimorgans you share as the average segment size, it's still probably solvable. And it goes down from there until you get to places like the one I just showed you, which was which is a tier E, which is 112 centimorgans, and the average centimorgan size is way below 12 centimorgans at 8.62. So this is a really great methodology for helping you to start sorting out your matches. And on the Medium post, Steve has a spreadsheet with the calculations already built into the spreadsheet. So you can enter data in and it'll do the math for you. An important caveat is that sometimes you only match someone with one very large segment of DNA. And so if you've got like a 40 centimorgan segment of DNA, a 50 centimorgan segment of DNA, it doesn't matter what the average segment size is, because that one large DNA segment means that it is solvable. You just have to figure out which ancestor you inherited that DNA segment from. So, rules of thumb for unknown matches. You want to look for matches that share at least one large DNA segment with you, which is a minimum of 20 centimorgans to 30 centimorgans long. If these matches also share at least 2 more segments that are 10 to 15 centimorgans long, you may be able to trace the match. You want to look for matches that also match your known relatives with large segments of DNA. Like if cousin Bob also shares a large chunk of DNA with this unknown match, you might match them on the same side of the family that cousin Bob is from. You want to keep in mind that you can inherit chunks of DNA from both sides of your family, which may make you look more closely related to a DNA match than you really are. Like you can inherit 60 centimorgans from your mom's side and 70 centimorgans from your dad's side. And in this one person you share it with, suddenly it looks like you share 130 centimorgans of DNA with them, and you do, but because they came from 2 different sides, it's a much more distant relationship. Analyzing endogamous DNA is mostly science, with a dash of intuition thrown in, and the more you do it and the more you look at your DNA matches and try to sort out the unknown ones, the better you get at figuring out when you shouldn't discount a gut feeling that may lead you to explore a match's family tree. And this is where we say there is no substitute for genealogical research, especially when you're dealing with an endogamous population. You have to build out the family trees for your DNA matches. You may be sitting there going, Jenny, why do I have to build out this family tree for, this person's cousin? But that may be the way you find out how they eventually connect to you. And it's actually really good practice because you're doing genealogical research for people who aren't your direct family, who might have different circumstances. And so you get exposed to record sets that you didn't always know that you needed to look at. Most people online do not have complete family trees, especially ones posted online. Some of them, like myself, have private ones, and I do that for my own reasons. You will need to do the genealogical research to build their tree for them, to figure out how your family connects to their family. And you should always remember while you're doing this to evaluate genealogical sources that you're using for things like the reliability of the person providing the information. If your relative, like my great-grandmother, dies in a hospital several counties away from where she lives, and there are no parents' names and they're written as unknown, that's because the doctor didn't actually know the parents' names, and the doctor is the one who filled out the death certificate. Whereas if it's somebody's wife who they've been married to for 75 years and they're the ones filling out the death certificate, the information may be somewhat more reliable. I am a firm believer that you can and should download your raw DNA data from any testing company that you've used so you have it for your own personal records. It's your DNA. A lot of companies will pop up a ton of warnings that you cannot keep it as secure as we can. Well, that's fine, but it's still your DNA and you have a right to own a copy of it, of the testing results. If you are looking for lost relatives or you're trying to solve an unknown parentage situation, it makes sense to upload your DNA to as many commercial testing companies as you can. The more databases you're in, the merrier. Ancestry does not allow DNA uploads, but you can upload your DNA data from Ancestry. You can download it from Ancestry and upload it to Family Tree DNA until very recently till MyHeritage, which may or may not be allowing uploads anymore. And you can definitely upload it to GEDmatch, and I'm pretty sure Living DNA still allows uploads. They usually charge a small upload fee to unlock features, but in general, this is less than the fee of buying a brand new DNA testing kit for each company. There are other tools to analyze DNA data that we have not touched on in this report, rather in this presentation, although many of them will be touched on in next month's presentation by genealogist Alec Ferretti. A key thing Ancestry does not have, which at this point I would give my right arm for, is a chromosome browser, which lets you view individual DNA segments on a chromosome map, like I showed you in the beginning, when comparing my DNA to my maternal grandmother's DNA. It's really wonderful for visualizing what segments of DNA you have inherited from which ancestors. With a chromosome browser, you can do amazing things like comparing DNA segments by chromosome to determine which ancestor you inherited the segment from, as well as triangulating DNA segments between two or more matches to determine if you are descended from a common ancestor. On some websites like GEDmatch, there are analytical tools to let you search by specific DNA segment, which is very useful. There are other tools that currently exist for analytical purposes, or things like auto clustering, which I refer to a little bit with the Leeds method, which does it automatically for you. Network graphs of shared DNA matches. There's two really good tools to test specific DNA match hypotheses, like if you sort of know how someone's related, but not 100%, called What Are the Odds Plus and Banyan DNA. And they let you enter in the amounts of DNA you share with various matches, and they run the probabilities for you. And also, being able to paint shared DNA segments on a chromosome map. And DNA Painter is my current favorite of all the tools out there, mostly because it lets me make a pretty map like the next slide. This is a portion of my DNA Painter map. The top blue chromosome is my paternal chromosome. The lower red chromosome on each set is my maternal chromosome. Every place there is a colored segment is a segment of my DNA that is assigned from a specific ancestor. You can see some of them overlap because some of them do overlap. Okay, additional resources for learning about interpreting your DNA results includes the International Society of Genetic Genealogy website, which has really great technical resources and also good tools. There's also blogs, books, journals, mailing lists, etc. I really love one of the blogs, which I adore, is the DNA Explained blog. It's written by Roberta Estes, who's a genetic genealogist and scientist. She's actually been working on the Million Mito Project to test mitochondrial DNA at Family Tree DNA. It has deep dives into topics like X-DNA inheritance, how to analyze your DNA matches on different databases and comparing them, and also how to use your DNA as evidence for proving genealogical relationships in your family. It also has another component to it because Roberta is of French-Canadian descent, so she has recently, over the last year or so, been profiling each of her French-Canadian ancestors, which has been fascinating because it's not an area that I particularly know very much about. There's also Kitty Cooper's blog, which has great answers to questions like, why don't we share more DNA with a sibling? How can the X chromosome help with maternal versus paternal? And also using Ashkenazi Jewish DNA to find family. Finally, Facebook groups. And I know it's Facebook, but it has a tremendous amount of resources on there in the common knowledge of people there. So there's the DNA Newbie Facebook group. There's also the Genetic Genealogy Tips and Techniques Facebook group. This is run by Blaine Bettinger, among others, who did the shared centimorgan project. He's a phenomenal teacher too. There's the Jewish DNA for Genetic Genealogy and Family Research group, which, as it says, is very much centered on Ashkenazi and Jewish DNA of all types. And I believe that is the end of my slideshow. So I will stop sharing my screen and return back to Zoom. And are there questions? Maria, would
Yeah.
you like to, should I just go through the questions one by one, the other ones, or would you like me to? Or do you want to read out the ones you think should be answered
One
first?
second. Uh, can you hear me?
I can absolutely hear you. Oh, I'll start with the 5 that are open, I guess. Okay. Robert Daly asks, how do you know that the gray pieces represent DNA inherited from your maternal grandfather and not one of your paternal grandparents? The answer to this is I do not 100% know this because my parents do share some small amount of DNA with each other. They show up as something like fifth cousins to each other, even though I have traced both of their family lines back 200 plus years for the most part on all branches. I have not found a genetic connection between them. However, because this is specific, the map I showed, let me see if I can put it up and share the screen with it up. Give me 1 second so I can refer to it. Okay. And back to sharing my screen for 2 seconds. Share. So the reason I know is this is specifically a comparison with my maternal grandmother. So when I created this chart, I said I only want to know what DNA I share with my maternal grandmother. And it's a fairly good guess that all the DNA that I might share with my maternal grandmother has to come from my mother. And so in this case, because it's comparing all the places where we match on, remember, we get 2 chromosomes from our parents. So it's essentially comparing all the places I match my maternal grandmother on my maternal chromosome, which is why there's only 1 line on this chart for each chromosome pair. And so the blue places are where I match my maternal grandmother, and the gray places are still my maternal chromosome, but the DNA is not identical. And because the DNA is not identical, it comes from my maternal grandfather instead. It's not to say there isn't overlap, because there probably is overlap, but it's small segments. Okay, let me stop sharing the screen again and see the next question. Okay. Mark Koppel says, I've read that Ashkenazi Jews started from a population of 6 women who married Jewish men as they passed through Central Europe. They then obviously multiplied within the community. So sort of yes, sort of no. What you're talking about is mitochondrial DNA, and it's a really interesting form of DNA. We have, so mitochondria are, if you remember like science videos from when you're a kid, are the powerhouse of the cell. And they are the sort of separate organism that lives within us that we've co-adopted into human cells over time. And it has an entirely separate genome than humans do. And if you test the 16,000 and change genetic locations on the genome, you can, on the mitochondrial genome, you can make sort of a phylogenetic tree that shows what maternal ancestor you are descended from. The other really neat thing about mitochondrial DNA is that it mutates very slowly. So if you match someone as an exact mitochondrial DNA match, and there's only 1 company that does that testing, which is Family Tree DNA currently, you are related within approximately 500 to 2,000 years with that person. And so what they mean by the population of 6 women is there are 4 mitochondrial haplogroups which are very prevalent among Ashkenazi Jews. One of them is, and they have recently redone the haplogroups, but the older ones are, it's a K1A something, it's N1B2, and there are 2 more who I have forgotten at this moment what they are. And so those are very prevalent, and they believe that the women who originated those haplogroups were originally from the Middle Eastern populations. And then there's a whole other bunch of haplogroups. Like I have in my family the W3A1 haplogroup, And that is absolutely descended from a Polish woman of some kind, who perhaps before they were Christian or afterwards intermarried into the Jewish population. Similarly, my haplogroup is a really rare one of, I think it's H5A1A. It has no hyphen. I'd have to print it out for you. But that one, I only match 8 people who are Jewish as exact matches, and everybody else is Christians descended from Czechoslovakia, basically. So it's really interesting that somewhere in the not so distant past, somebody married into my family and became Jewish, because as I can go back to 1760 and change on that line, and everybody was Jewish at that point. Can you recommend a webinar to build out a family tree? I'm looking for user-friendly info, safe from scammers to abuse the info, and of course, best for finding people. That's from Lynn Israel. I cannot recommend a specific one. I think there's a lot of really good basic genealogy webinars that are available online. Legacy Family Tree Webinars has a bunch. There's a number of them on YouTube that are free, including ones that the Center for Jewish History has done over the years. And what you would want is a basic genealogy webinar, which says, okay, I'm looking at something like a 1920 census, and the names are different from my family's names. Are they my family? And how do you go about analyzing, or are they this family I'm looking for? And it's how do you go about analyzing whether the information is accurate, whether this relative is really who they say they are, and so on. It's basically, it is basic traditional genealogy. And there's a lot of, also Family Search has a phenomenal amount of webinars they've done over the years through Roots Tech. And last I checked, it goes back at least 5 or 6 years. They've got indexed by topic. So you can go to Family Search and look for their videos. And that also will have a lot of really good basic genealogy introduction videos. Glenn Levy says, living DNA seems to be gaining popularity. They don't use Jewish reference populations for their ethnicity reports, which means they aren't very useful. I agree. They say I'm mostly Southern Italian. For shared matches, I've noticed their shared centimorgan totals can vary dramatically, excuse me, from other websites like Ancestry. Do you believe that living DNA shared centimorgan totals are trustworthy? I think they're trustworthy to a degree. I think it's, I would not use them as my sole source of comparison to find relatives. I would compare the shared centimorgan totals between various companies if you have access. And you can see that with some of them. MyHeritage, I love their chromosome browser to death, but they use imputation for their DNA matches. So sometimes what that means is they will take, say you have a small segment separated by a tiny gap where you don't match someone, separated by another small segment with another tiny gap where you don't match someone, and their algorithm goes, that should really be one segment, and makes it look like a longer DNA segment than it is. So each company uses different algorithms. They are computer science algorithms to some degree, and tries to analyze people's DNA, which is why I think you have to look at each website on its own and compare it to other companies. Linda McCracken says this is wonderful. Thank you, Linda. I appreciate the comment very much. Carol Sage Silverstein says, what does each segment mean? So each segment means it's literally an encoding of information for your cells to do different things based on which chromosome. The raw genetic data encodes genes, which is where we get the base word for genetics. And it's literally instructions saying how your body should function as an organism. Which company test should a pure, Paul Miller says, which company test should a pure Ashkenazi buy? I think in general, if you are looking to get the most bang for your buck, I would buy something like Ancestry, and then I would download the DNA and upload it other places. Particularly to GEDmatch, because that's a wide compilation of databases. I would also upload it to Family Tree DNA. I think Family Tree DNA excels at Y-DNA testing and mitochondrial DNA. Their Family Finder database, which is the autosomal one that people look at for relatives, is smaller than a lot of the other companies. But I think the things they specialize at, they do very, very well. So if you're looking, for example, to try to figure out who your Ashkenazi surname connects to, I would tell you to get Y-DNA testing done at Family Tree DNA, for example. But if you're looking to find the largest amount of cousins who your family has lost track of over the years, I would tell you to test an ancestor. Do the genetics companies, sorry, it's scrolling. Scott Norton says, do the genetics companies provide info on Ashkenazi associated inherited diseases? Example, Tay-Sachs, Gaucher, and dozens more. Sometimes. 23andMe does, with large caveats that they are not a medical testing company. Everybody else doesn't. There are websites that you can Google that will tell you how to look at your raw DNA data. I'm not remembering the exact name of them right now, but there are several, and you can search by specific, like say you read a research paper, and the research paper gives you the exact gene name. You can search by gene name and then like the testing company you tested at, and if that company has tested that portion of your DNA, because they're not doing your whole genome, because it would be cost prohibitive for any company to test your whole genome, then you will be able to at least look to see if you have that. And basically, if you think, the standard answer though is if you think you are at risk of a genetic associated disease, whether it's breast cancer or Tay-Sachs or even a propensity to like high cholesterol, you should consult with your doctor and you should get medical genetic testing, which is the only type of genetic testing that an insurance company will accept in the United States, among others. Daniel Scharf says, can you explain how DNA can be used to trace ancestors back more than 100 years since no DNA then? Everybody has had DNA. Basically, since humans started walking on two legs in Africa, we have had DNA. Animals have DNA. Your cats have DNA. Your plants have DNA that you grow on your windowsill. There's no DNA testing done 100 years ago. But all of those people who were your ancestors have passed on tiny bits of, have passed on DNA segments to you. So, excuse me. So you can match those segments to known cousins who may have also inherited those same segments. Anonymous attendee, hello anonymous. My parents don't have half siblings, yet Ancestry uses that term in predicting relationships. Yes, they tried really hard to predict an accurate relationship. It doesn't always happen. Is there a, excuse me. Vladimir Malinsky says, is there a clear shared segment between all Jewish DNAs? No. No, there's not. There's what's known as overlap segments, which happen among people of all populations. But the overlap segments are just areas where the same DNA we build up. Give me 1 second to go grab a glass of water. I do apologize, or I'm not going to be able to talk to answer everybody else's questions. I'll be right back. Thank you for the brief interruption for getting water. Okay. Barry Clark says, realistically, how many hours would a relatively alert senior need to learn enough about Jewish DNA to be able to work effectively on our DNA results and our cousin's DNA we manage? I mean, to be realistic, hours. You're not going to analyze it in an hour. You need to do the genealogy behind every match who you think you might be related to, and basically build out the family trees. I know you say you're, oh goodness, it rolled. Sorry about that. But anyway, You can't analyze Jewish DNA without building out the family trees. It doesn't work. Glenn Levy has another question. Thank you, Glenn, for saying you like the presentation. If I can ask another question, what do you think about ancient DNA sites like Illustrative DNA? They claim to be able to show the ancestral components of your ethnic breakdown based on G25 coordinates. Do you find the science reliable? I find the science fascinating because it's using population genetics, but I also would not take it as 100% reliable because it's trying to match you to really deep populations of ancestry based on autosomal segments. And most of our autosomal segments are not more than like 300 years old. So it's doing sort of the same thing that ethnicity estimates are doing. It does work a little bit better for non-endogamous populations. MyHeritage has one now, which I'm forgetting their marketing name for it, which tries to show you the population genetics graph. It works a little bit better for my husband's non-Jewish family than it does for my family. But even then, like, it's taking its estimated best guesses. I would, if you feel up to it, I think it's more useful to read more current science papers that are being done by scientists in the field who are doing things like analyzing ancient bones that are found and trying to find where they fit within different DNA populations to try to track the path of human migration, for example. I think that's more accurate because they're doing it with more reliable science than a commercial website. Do we have any more questions? And I apologize for losing my voice in the middle of this. There is another question. Okay. Days and hours to understand how to. Okay. So Barry Clark says, it seems there is so much to learn. There is a lot to learn. I am still learning. My question is how long it takes in hours to understand how to analyze DNA results, not the work once we understand it. I think it takes however many hours you're willing to put into it, which I know is an answer, but that's not what you're looking for. But I think it's how fast you educate yourself on it. I think webinars are great. I love watching webinars. I sit there folding laundry at home watching genealogy webinars. I would not say it would take 100 plus. I would think of it like you're taking not a college course because it's not a college course level, but you're taking a deep dive into a topic to try to learn how to analyze it, and you want to feel comfortable with it in order to be able to apply it to your DNA matches. So I would say at least 10 to 20 hours, but it really depends on the person. And I think you learn more by doing and actually trying to analyze your DNA matches than you do by just reading books. And I mean, thank you for all coming to this lecture, but I think also this is the kind of thing that putting it into practice is really effective. And even if you analyze something and then you come back and you look at it a year later and you go, wait, I see something totally different, that's important because it means your analysis skills have evolved through a year of doing it. Oh, thank you, Barry. And everybody else who has sent such nice comments. Thank you very much. Okay. Daniel says this all seems quite complicated. It is complicated. Can a non-professional try to do this work? Yes, absolutely, 100% you can. Would hiring a professional make more sense? It really depends on your patience and your budget. You will get faster results by hiring a professional, but also, at least to me, it takes away from the fun of doing it yourself. Where would I find a genetic researcher specializing in Ashkenazi Jews? The standard answer is the Association of Professional Genealogists has a really wonderful searchable directory. It's apgen.org, I believe. And you can search by Ashkenazi and by DNA. or combinations of keywords. And so then there will be a bunch of genealogists you can contact and sort of oh yes, see, my colleague has put it in the chat. The URL, my standard caveat that I tell everybody hiring a professional as genealogist is a service. Get several price quotes from different genealogists. Do not go with the exact first one you see, because you're shopping around and you're trying to find the best service provider for what you're doing. And you may really, vibe with one genealogist and you may really like their approach, and you may really not do that with another one, even if that other genealogist might be a little less expensive. So it definitely pays to talk to every single one you're thinking of working with. Vladimir, so there was nothing. What links Sephardim and Mizrahi and Ashkenazi Jews via DNA segments? So that's not the topic of this talk, unfortunately. There will be a Michael Wass, who is a wonderful genealogist, is giving a talk on Sephardic DNA later this year in August. It'll be in person, but also live streamed on Zoom. So if you're in the New York area, you can come to it at the Center for Jewish History. We don't have the ticket information up for it just yet. We will soon. And you can really see the relationship between Sephardim, Mizrahi, and Ashkenazi Jews if you're looking at the paternal lines through Y-DNA. There are several Y-DNA haplogroups which are very strictly Sephardic, but then there are others that are sort of interlinked to Ashkenazi, and you can see where they split apart at certain points. Bennett Greenspan is a speaker who has given a really interesting discussion about how if you go far enough back in his family, all of a sudden you stop hitting Ashkenazi Jewish matches from the Ukraine-Poland area, but you start finding he matches people from Catholic descent from Spain and Mexico. So it is very fascinating. Robert Daly says, before today's presentation, I knew that Ashkenazi Jews are from Central and Eastern Europe. The slide that discussed where Ashkenazi Jews originated, but it went by too fast. I'm curious to know about the connection to the Levant. We don't 100% know the connection to the Levant. We know it exists because we can see it in DNA. And it's actually fascinating from a population genetic standpoint. We Ashkenazi Jews and Sephardic and Mizrahi Jews match very similarly to Samaritan populations and other Canaanite populations that are still in the Middle East today. But we do not know the exact path that Jews took. We know there was a diaspora. We know there was the Bar Kokhba revolt in the 150s, 150 Common Era. We know that Jews were enslaved by various cultures, such as the Roman Empire. We know that Jews ended up in various different locations during diasporas. Even earlier than that, we know that Jews were taken to Babylon as slaves. So all of that has intermixing that goes into being Jewish around the world, basically. The current best theory, which is why I was explaining before with the living DNA, yes, it says I'm Southern Italian, is that Southern Italian genetic population genetics looks really similar to Ashkenazi Jewish population genetics in certain ways, where they've got Middle Eastern heritage and they have European heritage intermixed. And it's gotten a little bit more refined over the years with many companies, but if you go onto GEDmatch and use some of their much older, and by older, I mean like 15 years old, so it's not that much older, population analysis tools, it'll tell you you're Southern Italian, because that is the matching reference population that you match most closely to at that point in time when they did that. It is 6:09. Are there any other questions, or we could end early if people wanted? Okay, Robert has a question about DNA tests. Oh, thank you, Vladimir. I think if nobody else has any questions other than Robert's question about DNA tests will be the last one. Just waiting for it to appear in my screen. Glenn has a question. My maternal haplogroup is K1A4A. Is that typically European or Middle Eastern? So there's a really great book by Kevin Allen Brooks called The Maternal Lineages of Ashkenazi Jews, where he tried to put together as much data as he could from various sources. And he sort of breaks down to the best of his knowledge where each maternal haplogroup originated. So I know it's available online as an ebook. I know I have a Kindle copy of it, but you can also buy a physical edition of it from various online book retailers. I think it was about $23 when I bought it. And it's really, I find it fascinating, even though it's about 2 years out of date. But the great thing about it is that he has an ongoing database. If you're familiar with the Jewish Gen Mail boards, the discussion boards, and every now and then he posts updated information about new samples for mitochondrial lineages for maternal haplogroups, so that that is refining his estimates of where they come from. How do I get my 23andMe data and what do I do with it? If you have tested with 23andMe, within the settings, you have to poke around for it. There will be a spot that says download raw data. They will tell you they need something like 24 hours, and then they will send you a link to download it. Once you download your 23andMe data, you can definitely upload it for free to GEDmatch. You want to make an account. You'll have to register and make an account for them. You can, I believe Family Tree DNA is once again taking uploads of 23andMe data. I do not know if any other company is. I know it is still available. You can upload it to GEDmatch, which is not the world's largest DNA database, but is still somewhere else that you can look. I will just keep talking while we wait for Robert's question. In general, on the subject of 23andMe, it is disappointing all the woes the company has gone through, because there are people who tested only on 23andMe, and a lot of people have deleted their DNA data from there. But I think it's a very personal decision of anybody who has tested whether they would like to keep it on that company's database and whether they trust that company. And I can't advise one way or another because it's your genetic information. Okay, I am still waiting for Robert's question. There we go. Sorry. Robert says, and this is the last question, as I build my family tree, I am connecting with cousins I've never met. Assuming that none of the cousins groups that I'm in touch with have done DNA testing, would it make sense for all of us to take the same DNA database test? Yes, 100%. GEDmatch does not have a test. I know you included it. It's more of like it allows you to compare results from different databases to each other. Ancestry or Family Tree DNA, I would recommend testing all on one if you can, because it lets you compare how much you share with each other too. And especially with cousins, if you want to know how much you share with cousins. And they're willing to take a DNA test. And once again, that's a personal discussion for other people. Karen asks, my sister and I relate to people differently. Me as second and she as third cousin. Does this just relate to different segments from different parents? It just means you inherit, assuming you and your sister are full sisters, meaning you share both the same mother and the same father. It just means you inherited different bits of your parents' DNA, and your cousin, the people you relate to, also inherited different segments of DNA from their parents. That's the element of randomness, and it's just when the person was conceived, it's what combined. Yeah, so I'm going to wrap up now. I hope all of us will join us. I hope all of you will join us next month. I am moderating next month, not presenting. We have Alec Ferretti, who is a fabulous genealogist, who is going to talk about more advanced strategies of analyzing Ashkenazi Jewish endogamous DNA. I love his presentation. It's very humorous at certain points. And so that's on July 7th, and the ticket link is up on the Center for Jewish History. Michael Loss will be speaking about Sephardic DNA in August, on August 3rd in person. Coming up, we have Michael Leclerc at the end of September, who's going to give a presentation, not a DNA-based one, about researching your LGBTQ ancestors and how to do that. And we have Raquel Levy-Toledano will be presenting in November on some element of Sephardic research. We're still nailing it down. And then we have Gil Bardij in December, who will be presenting on a DNA case study, among other things, showing you how you can combine documents that you find in traditional genealogy along with DNA evidence. And I thank you all for coming and asking such amazing questions. And I am going to end the meeting now. Thank you.